raise your hand before you speak or if you want to say something. So, are we all agreed to follow these rules? Yes. Okay. So, let's start our discussion by answering a simple question. So, what are some things you do when you prepare for an upcoming examination? Um, yes, Dom. That's right, you review. Any other hands? Yes, Aliko. Prepare the materials for going to reduce the taking time. Very good. Prepare the materials. Or any other hands? Friends of that? Yes, Jason?
Memorizing the topics.
Okay, very good. So when your exam is nearing, diba, you review, you prepare your materials, and also you memorize the concept of your subject. So why do you think it is important to do these things or to prepare? Yes, can so we get higher grades on the higher exam. Okay, very good. So that we can get a higher grade. So let's have another situation. Every morning, when you go to school, what are some things that you do every morning? Yes, Jason? Fixing the bed. Very good. Any other hand? Yes, Katniss? Eating breakfast. That's right. Shan? What? Walking. Walking to school? Okay. So, yes, taking a shower or taking a bath. So, why is it important to do these things before going to school or before you leave your house? Yes. For hygiene or for you to become ready, presentable or ready to school. So, if we humans prepare before something happens, do you think cell also prepare before cell division? Yes. Why do you say so? Says yes. Yes. Okay, right, then I heard you. Do you think cell also prepare before cell division? Yes. So just like us, cell also prepare before cell division. So So before the cell divides, it needs to pass through an important stage, and that stage is what we call the interphase stage. Can you say it with me? Interphase stage. Very good. And that interphase stage is what we call the preparation stage of the cell. So before that, we have here the cell cycle. So when you hear the word cell cycle, what comes to your mind? Cycle. Yes, Akiko. Going and continuous. That's right. It's like a continuously. So if we combine the two words, cell cycle, what do you think that means? Cell cycle. Yes, Sean, we have opinion on that. Okay, so cell cycle is a series of events that cell goes through before it grows and divide. And it is divided into two stages. The first one is the interphase stage. What is the interphase stage again? Ulit. Again? Preparation stage. you still remember what is the function of the nucleus? Yes, Jason?
It controls all functional activities.
Okay, very good. The nucleus controls the cell. So, the nucleus plays a vital role in cell division because this is where the processes this is where the processes takes place, and it controls the cell and keeps the DNA inside. So before that, we have the interphase stage. So as I said a while ago, this is the stage where the cell will pass through before it goes through or pass through cell division. So in interphase stage, we have three subphases. So the first one is G1 phase or the GAP1 phase. The second one is the S phase or the synthesis phase. And the third one is GAP2 phase, the G2 phase or the GAP2 phase. So before that, this chart shows the interphase stage of the cell cycle, and each color represent a specific phase. So as we go along, we'll discover what might happen or what event takes place in each phase. So let's start with the one phase. So what do you think will happen in this phase? Do you have any idea? Since this is the beginning of preparation stage of the cell. Do you have any idea? None. So in that one phase, the first thing that the cell will do is, can we read together?
Cells grow inside.
The cell grows inside, or they become bigger. So why do you think the cell grows inside? Or why does it need for them to become bigger? Yes, Sean.
According to it takes a big deal for the divide
So you have a point. So the cell grows inside in preparation for the DNA replication. But not just that. They also make protein. So the cell also makes protein. So what do you think is the importance of this? Why is it necessary for them to make protein? So that yes, yes. You have a point. Thank you for sharing it again, young man. So the protein acts like a support for the cell that they will use when they divide later. So what do you observe in this? What is this? This one. There is at the end of this phase, we have the checkpoint. So what do you think is the importance of this checkpoint? Why is it there? Before proceeding to the next phase, why is there a checkpoint? Yes, Rachel. Saying something.
It separates the G1 phase and the S phase so that the cells that are still in the G1 phase have been prepared to
Okay, you have a point, but the checkpoint makes sure that the cell is ready before entering the, what's this? The S phase. So, what
Dahil hindi ko po sila ready, nakapila ko sila sa mga
Okay, that's right. So it will not pass through or proceed to the next phase because it will not pass through to the next or it will not proceed to the S phase, but it will repair itself. You get it? It will repair itself first, or it will wait and repair itself first. So after that, can someone decide what's happening in the G1 phase or GAP1 phase again? May I call someone? Yes, can you?
Cells build in size, makes protein, and preparation for DNA replication.
Very good. In GAP1 phase, the cell grows in size. And because it is the preparation for DNA replication. And also the cell makes protein. And after that, it will not proceed to the S phase or the synthesis phase. So when you hear the word synthesis, what do you think does it mean? Synthesis. Do you have any idea? Yes, Dom? Synthesis. Okay, I call someone. Yes, I will. But after your mind, you read the word size. What? Sizing? Actually have to make. Because in every phase, synthesis means make something new. So what do you think is this something new that the cell needs to do? What's this make something new? What's this something new that the cell needs to do? What do you think is synthesis means? Yes, Raisa. Nice try. Any other hands? So synthesis means to make something new. So in this phase, the cell duplicates its DNA. So we have the DNA is duplicated. So in this phase, the DNA is replicated so that the chromosome is also replicated. So after the DNA is stacked in the S phase, each chromosomes can now form two identical halves. Now, look at this picture. Can you describe its shape? Can you describe its shape? Anyone? Yes, Akito? Yes, that's correct. And it is like a elongated, and this elongated shape is what we call the chromatid. And chromatid is one of the two identical halves of a replicated chromosomes. Now, let's look at this second picture. What can you think? What can you think if this one chromatid is being paired up with its identical copy? What do you think will happen? Okay? What? And what? They form with what? What did they form? Yes, they have. Yes, that's correct. They form a chromatid are two identical chromatids that are stuck together. Now, I want someone to differentiate what is chromatid and sister chromatid. Anyone? Anyone from the class? Yes, you want? Very good. Now, let's proceed to the checkpoint in the space. In this checkpoint, the cell checks if the DNA has been completely and correctly copied. Then, if the DNA is now completely and correctly copied, it can now enter to liquid phase. Now, here's my question. If human phase is the cell grow in size, and how about in liquid phase? By looking at this picture. What do you think will happen in the cell? Yes? Kenzie? Yes? You have a point. How about the others? Thank you, Kenzie. Yes, Andre? Yes, that's correct. In this phase, the cell keeps in this phase, the cells keeps on growing. The cells keeps on growing. Now, let's proceed to the checkpoint. Let's proceed to the checkpoint of liquid phase, where it detects and repairs the DNA damage, ensuring the cell is ready to proceed to end phase. Again, here's my question. Why do you think it is need the cell to completely copied before it enters to end phase or the cell division? Anyone? Yes?
Not only for the individual other cell, but also be compatible.
Okay, that's correct. Now, that we know the three phases, based on our discussion a while ago, can you guess how many hours does the human spend out of 24 hours? For human cycle, it takes about 24 hours to complete the cell cycle. Out of those 24 hours, 23 hours is for the interphase. Now, based on this chart, can you guess how many hours did the human face? Guess any hours. If this complete cycle, if this cycle can take 24 hours to complete. Yes, you want? Can you guess how many hours? 16. Nice try. How about Gerson? Thank you.
For the genome,
Yes. How many hours? 11 hours. Nice try. This is Gerson. Yes, Kenzie.
Yes,
That's correct. That's correct. The human phase takes about 11 hours to complete this phase. How about in S phase? Can you guess how many hours does it take? Yes, Ryzen, 6 hours. 6 hours. Nice try. Yes, Ivan. Nice try, Ivan. Yes, Althea. 8 hours, that's correct. In S phase, it takes about 8 hours to complete this phase. How about in G2 phase? Can you guess how many hours? Yes, Joanna? 4 hours. Nice try. Yes, you want? Nice try, thank you. Yes, Zam? Because that's final. Nice try. I think that's 4 hours. Okay. Yes, Jason?
5 hours to 5 hours to 5 days.
Nice try. Yes, Kenzie? 4 hours, that's correct. Zero phase takes about on how the bacteria can quickly spread. And one. Yes, right. Yes, you have a point. And one. Do you think there is a connection on how the bacteria can complete its cycle in just 20 minutes on how the disease can quickly spread? Just like this. And one. I will, since no one wants to answer, I will go on. Yes, Akiko? That's correct. The cells and the bacteria can grow quickly. They can grow quickly because it just takes 20 minutes to complete the cell cycle. And that's why you need to wash your hands and take medicine if it is needed to stop bacteria from spreading. And for the rest is the membrane. But this part is for the cell division. Now, let's have an activity. Now I have an activity, and this activity you will use a clay. And I will group you into three groups. This will be the group 1, group 2, and group 3. Each group will have a clay and a activity board. So in this activity board, you will paste the output of G1 phase, S phase, and G2 phase. And then you will write what happens in G1 phase, S phase, and G2 phase. And at the back of the activity board, you will answer the question. And I will give you 10 minutes to answer that. Then after, I will call a representative from each group to come here in front and show their output. Do you understand, class? Okay. You may now form a circle. You will
Kasi nung clips,
Last one, you have 5 minutes to finish that.
Isa pa kasi nagtibay ko kasi.
One minute